Welcome back, everybody. I hope you had a little break there. As mentioned before, what we're going to be discussing in this second section is the idea of how we go from that single fused cell called a zygote to the complex individual that we become by the time of birth. And when we're looking at that particular topic, this growth, what we often do as psychologists and as biologists as well is try to describe this growth in three different sections. Those sections are the germinal period, the embryonic period, and the fetal period. The germinal period is by far the shortest of the different periods of growth that are out there and lasts for about 2 weeks. And in this class, we're going to highlight all of the things that happen during the germinal period and also detail why we call it the germinal period. Second period, being the embryonic period, follows that germinal period, and again we're going to focus on what happens during that period and also to explain why it's called the embryonic period. And the final one, that fetal period, where most of the growth and development actually happens within the womb of the mother, is another thing that we'll be highlighting and discussing throughout this class. Now, as we mentioned earlier, multiple times actually, when we look at birth and growth, what we see is that first conception allows people to actually create that zygote. So the merging of those two germ cells allows a person to become that one full cell with all of the DNA that's required for growth. But obviously, that has to multiply. And in this germinal period, for those first 2 weeks, the process of multiplication that we call mitosis is occurring very rapidly. We go from actually about one cell, well, from exactly one cell, that zygote, to approximately 100 cells by the first week. That means that these cells are multiplying by the day. In fact, that first cell, usually in the very first day, has already reproduced either 1 or 2 times to get to that magic number of 100. And when we get to those 100 cells, we continue rapidly multiplying and get almost to 1,000 cells by the time we've actually finished this germinal period. And with that cell multiplication, not only do we have a large number of cells that are starting to be produced, but we also start to have some diversification. Much less than we'll see later, but this first diversification happens in order to allow us to survive within the womb. In particular, what happens is our cells multiply into either becoming something that we call blastocysts or something that we call trophoblasts. The blastocysts are the cells that will eventually develop into us. So develop into the embryo, which eventually develops into the human being that we will eventually become. The trophoblasts, though, are a collection of cells that kind of form an outer layer that's going to protect the blastocysts. And this not only protects the inner cells from the outer cells and the outer environment that the child is going to be exposed to, but it also creates that connection system that's eventually going to provide nutrients for those inner cells, for those blastocysts. Now, this germinal period is highly marked by cell multiplication. There's not a ton of diversification except for the blastocysts and trophoblasts, but it lasts for only about 2 weeks. And that kind of multiplication is stopped when, or I guess not the multiplication is stopped, but this particular stage of development is kind of marked at its end when something that is called implantation occurs. And this is when that cell, actually that collection of cells at this point, attaches to the uterine wall of the female. And once that attachment happens, we move on to the next stage of development called the embryonic period. Now, before we move on to this embryonic period, though, there's 2 interesting facts that I'd like to point out, things that aren't usually mentioned in the developmental class. One of the interesting facts that your book actually talks about is the fact that this implantation process, which seems rather automatic, and when we see videos of it, you see this perceptual thing where the cell is eventually going to float down and just automatically attach to the uterine wall of the female. Well, that process is not nearly as simple as it seems. In fact, usually the cell first implants or attaches to the female's uterine wall between 7 to 10 days after ovulation. However, there's a lot of different things that can occur along the way, so that cell can actually become dislodged, or those collections of cells can become dislodged very easily. And not until about 10 to 14 days after ovulation does that cell firmly embed, or the collection of cells, firmly embed itself into the female's uterine. And in all reality, a lot of these fertilized cells, these kind of complex beings that are starting to form, don't actually implant correctly. In fact, about half of all fertilized eggs will not implant in the female's uterine and will not be carried to term. And what's even more interesting is once we get into this next stage, the embryonic stage, as the cells are multiplying, a number of them will not develop correctly, which means that only about 1 in 4 of all fertilized cells will actually come to term, well, at least come to the point where they'll actually progress fully through this next stage that we call the embryonic stage of development. What is the embryonic stage of development? Well, once that fertilized cell, or I guess collection of fertilized cells attached to the uterine wall, what we see is that this kind of attachment sets off a cascade of events that occur in the developmental process. One of the big things that starts to happen is, even though these original cells are multiplying very quickly in the beginning, they start to multiply even faster. And not only do they multiply faster, but they start to create very specific cells. We call this organogenesis, or organ formation within the system. And those organogenesis that occur, sorry, I'm not sure the correct term there, but those developments that occur actually allow the system to develop a number of distinct characteristics. One of the first things that happens is that there's a creation of something that we call a life support system, where we have an amniotic fluid that starts to be formed from some of these cells that are multiplying, an umbilical cord that connects that embryo, which eventually will become the child or the fetus to the mother, and we've got something called a placenta, or a chorion, which actually is a protective barrier for the child from the mother's internal organs and the mother's internal system. And this life support system forms very quickly in this particular process, in this embryonic period. But in addition to that, there's a number of other systems that are also forming. So you don't just have this massive blobs, or a massive blob of cells that's actually forming. Now we have a collection of different types of cells that are starting to form within the individual. One of the collection of cells is something called the mesoderm, which creates our circulatory system, our bones, muscles, and our excretory and reproductive systems. And all of these things are actually forming, or starting to form, in the embryonic period. In addition to that, we have another set of cells that we call the endoderm cells, which will eventually form our digestive and respiratory systems. And we've got a third different type of cells that we call our ectoderm cells, which are responsible for creating, this is very important for psychology, our nervous system and our sensory receptors. So this is what's going to allow us to have those thoughts, those processes, and be able to make sense of our environment in the long run. If we look even closer at this last system, what we see is this particular portion is sometimes called a neural tube. In fact, those collections of cells that form this system are what's going to eventually look like, as you see in the picture, a long strand that goes from the top of the head to the tail of the individual, or the hind bone of the individual. This starts to form about 2 to 3 weeks after conception, or almost immediately after implantation, and this neural tube continues to grow. And the cells that are there are going to develop into something that we call neurons. And these neurons allow our brain to eventually form and to communicate with the rest of our system. And as this embryo is growing, this nervous system, or these neural tubes, are actually open, and they're continuing to grow as the embryo continues to expand. However, eventually, this neural tube has to close. It has to close on both the top and it has to close on the bottom, because once it closes on the top and the bottom, then our specific nervous system functions can actually form. So the top region becomes our brain and our brainstem and other systems that are connected to that, and the lower region becomes our spinal cord and all the different neurons that travel to the rest of our body in order to control our motor and sensory systems. If this doesn't close, this neural tube doesn't close at the correct time, we can actually experience some major birth defects. The first birth defect is something called encephaly, and this particular defect is when the top portion of the neural tube doesn't close. And when that top portion doesn't close, unfortunately, the chances of survival for this eventual fetus are almost zero. Encephaly is not something that usually allows a fetus to be carried to term, because if this doesn't close, then the brain doesn't form fully, and a lot of the basic functioning is also not going to form. So very few, if any, children ever survive if encephaly does actually occur, and if they do actually live to birth, most of these children die, well, almost all of these children die within a matter of days, and all of them will not make it past maybe a year at most if this particular tube does not close in that spot. The other end, though, the other region of the neural tube that also has to close, the bottom one, if that doesn't close, what happens is something that we call spina bifida. And this is something that's dependent not upon whether or not the neural tube exactly closes, but when it actually closes. And if the lower region closes on time, most of us don't have any problems, and this happens for almost everybody. But if the lower region does not close the correct amount of time, what we can have is developmental issues based on when this tube actually closes. So if you experience spina bifida, but your neural tube eventually closes, you could actually just be paralyzed, say, from the legs down or from the waist down. And there's actually a number of people that have survived throughout their entire life with spina bifida, just kind of this failure of this neural region to close at the right time. And they've all suffered from varying degrees of paralysis based on when we were able to actually close this neural tube and get the system working up to the point where there wasn't any malfunction. One other interesting fact, which is something that's very kind of surprising to a lot of people, is that by the time we reach the end of this embryonic period, even though there's still a lot of development that actually has to take place, all of our major organs, all of our major systems have already started to form. So that means by about week 8, even though our fingers and our hands and everything else still has a lot of formation to go, most of our major systems have already started to diversify, have already started to build into that person that we're going to eventually become. After about 8 weeks, we move into the third period of development that we call the fetal period. And this is after the embryo has actually formed all the critical organs. And now the fetus is, instead of creating organs, starting to refine those organs. So when we talk about the fetal period, what we're talking about is a stage of fetal refinement, not fetal formation. Now, this fetal period is usually broken down a little bit further. We talked about this earlier when we discussed this concept of the trimesters of development. Well, the fetal period actually takes place near the end of the first trimester, and it's very often broken into 4-week portions, because during these portions, different types of organs start to perfect themselves. And these organs kind of come in stages. So in specific stages we might have one organ developing, and then that organ might not develop again for weeks, or maybe never again, until after birth. The first trimester, though, the first trimester lasts up to 12 weeks, which means the first 4 weeks of the fetal period is actually hallmarked by the first, or I guess contained within the first trimester. And in those first 4 weeks, specific things are starting to be refined. In particular, our extremities are very important and very refined in this particular period. So our arms, legs, fingers, and toes not only start to form, but the nervous system that actually connects those arms, legs, fingers, and toes to our brain are starting to form. And that allows us, not extremely intentionally, but it allows us, when inside the womb, to actually move these organs. If we focus in on the face, even though the emotional expression is not something that's really quite there, there's no idea of whether or not a smile or frown means anything, the child also starts to develop the nervous system capabilities to do some of these basic facial structures and some of the basic responses, the basic, what we'll call reflexes, of swallowing and sucking. Now, we can also start to see at this stage that we can actually distinguish sex. And by the time we get to 8 weeks and move on, we're talking about now a fetus, not an embryo. In the second trimester, if we break that down into 4-week periods again, we see more growth. So we see continual growth of organs like the heart and skin. In fact, we can even see fingernails and toenails by the end of 16 weeks. But more importantly, and this is something that we study in the psychology class, we also see that there's refinement in our motor system. In fact, children by this time, up to 16 weeks, are actually able to start to coordinate their movements. So if something actually occurs, if they feel something, or something is going on, they can actually move a specific limb. It's no longer just a kind of random reaction. There's actually an intention behind some of the movement that these children are actually doing. As we move on, we get continual growth. So we see a lot of the specific systems actually forming by the time we're 20 weeks old. And we're actually much, much bigger by the time we reach 20 weeks old. In fact, we've doubled in size over 4 weeks. And we start to be able to do some very basic kind of reactions. We're going to call these basic reactions reflexes. One of them is something where we suck our thumb, and you can actually see, and this is kind of unusual, but you can see a fetus actually sucking its thumb in the mother's uterus. And you can also see that a child sometimes might even start to hiccup. This is a very basic reflex that sticks with us throughout our lives in order to kind of prevent certain things from happening. But these basic reactions are not only interesting, but they show us how diversified the nervous system has actually been by the time we reach 20 weeks. If we move on to 24 weeks, and the fetus is still doing well, we start to see a lot of really critical basic functions actually occur for these young infants. One of the things that starts to happen is they develop another reflex that we'll call the palmar grasp eventually, where they can actually grasp things very firmly. And at this point in time, because most of the major organs have formed enough, this is the first point where a fetus could theoretically survive outside of the mother's womb. And that doesn't mean that all fetuses will survive if they're born at 24 weeks, or if a mother has to deliver at 24 weeks because of some specific issue. But it is the first point in time where a fetus actually has a chance of surviving outside of the mother's womb. When we move on to the third trimester, we see more growth. So at 28 weeks, we see a large amount of activity, and we see one of the major, very critical functions of reflexes, and that's rudimentary breathing, also starting to form within these children. Now, they're not actually breathing in oxygen, but their lungs are starting to kind of develop, and the system that allows them to breathe, allows them to actually do what's critical, starts to form at about 28 weeks. At 32 weeks, and we're going to talk about this in the next section, our sensory systems start to really diversify. So children start to have stages of wakefulness and sleep, which is a whole different form of consciousness, and that shows us that the brain has developed significantly by this point in time. But we also see that children start to be able to not only coordinate physical movement, but they start to be able to really interact with their environment. So they can actually respond physically to sounds. They can move to specific sounds, or if they like something, you'll actually have infants, well, I guess at this time we'll call them fetuses, actually do things like kick or move in response to something that's going on in the environment. And this particular responsiveness is something that's very interesting. Now, your book talks about the fact that this might actually occur a little bit earlier. But we do know with almost absolute certainty that by 32 weeks, almost all infants are capable of responding to this sound. When we move into the last couple weeks, say 36 to 38 weeks, we actually see a slowing down of the physical growth and a slowing down of kind of all the changes are going within the infant. And instead what we see is kind of a rapid growth that prepares the infant really for some of the interactions that it's going to have, in particular for that birth that's going to occur very quickly. So instead of a constant movement, the reaction, the child is now kind of in a preparatory stage for the actual birth. Now, in addition to this physical growth, especially in the later stages of development, what we also have to be cognizant of is the fact that the nervous system within individuals is constantly growing. We've mentioned some of the basic motor and sensory abilities that children have, but in order to do that, what we have to see and what has to occur is growth within the nervous system. Now, we've already mentioned this idea of something that we called a neural tube, and this was something that starts to form actually in the embryonic period, and it closes at some point in time during the fetal period. In addition to having that neural tube close, other very critical changes are needed for a person to develop a very coordinated and normal nervous system. In particular, two types of growth have to occur, and both these growths occur on the cells that are a part of our nervous system and a part of that neural tube. We've already used this name before, but just to define them, what we're talking about here with these cells is something that we call neurons. And these are cells that are specifically designed for this communication that allows us to do all of those coordinated movements and behaviors that we think of when we talk about things like thoughts or reactions or interchanges with the environment. These neurons that we have contained within our neural tube, once it's closed, must do two different things in order for our nervous system to actually develop fully. One of the first things that they need to do very quickly is form massive numbers of neurons within our system within a very short period of time. And we call this massive reproduction of these cells neurogenesis. And when we're growing, when we're developing within the womb, neurogenesis is something that's very, very rapid. So you've got cells multiplying at unbelievably quick rates. And these neurons not only are multiplying, kind of creating intricate systems that will eventually become our brain and our nervous system, but we also have these cells moving in very specific paths. And we call these paths, or this movement, neuronal migration. And this neuronal migration actually allows our cells that are multiplying constantly to move into places that allows us to do all of the coordinated movements and eventual thoughts that we're capable of doing. So instead of just multiplying in specific areas, what we have is numbers of cells multiplying and then moving out into our different organs and moving into different collections throughout what's going to eventually become our brain in order to create that very, very complex nervous system that we actually possess. And once that movement is actually formed, what these neurons will do is actually almost connect to each other. So they create a very, very small gap between each of these cells that we call synapses that allow these cells to eventually communicate with each other. So as we're growing physically, we're also growing in our neurons and in our nervous system. And that growth in our nervous system and in our neurons allows us to eventually do all of those dramatic things that we're capable of doing throughout our life. At birth, when we're looking at a young child, what we see is that these children actually, when they're born, have as many neurons at birth as they're ever going to have throughout their entire life. In fact, these children at birth are born with somewhere between 60 and 100 billion neurons in their brain and nervous system. This huge onslaught of neurons allows a child to really be able to adapt to the environment that they find themselves in and really develop all the different skills that are going to be necessary for them at some point in time in their life. These neurons will not grow nearly as much. In fact, this concept of neurogenesis is something that's being greatly debated within the field today as to whether or not we ever have more neurons formed. But we know for sure that these neurons will not grow nearly to the same extent as a child has had growing inside the mother's womb. And in fact, most of the time, what's going to happen, we're going to talk about this later, is those neurons will very often die away and get smaller as we progress throughout our life. But that's for another class. In addition to looking at neurons, though, what we can also see is that the motor system of these young children is very limited at birth. In fact, even though there's some coordination of movement, there's an idea that if a child wants to move a leg, it can. That coordination of movement is extremely limited. And when you pair that with the fact that the musculatory systems of these children is very weak at birth, what you see is newborn infants have almost no ability to really control how they're interacting with their environment. So when you see a young baby, what you'll often see is parents kind of cradling the neck of a child, and that's primarily because on its own, a newborn infant's not actually capable of holding up its own head. Now, in addition to that poor motor system, they also have very poor sensory systems as well. So vision is absolutely horrible in young children, and things like smell and taste are things that we believe really haven't formed for quite some time, and won't form for quite some time within these young infants. The only sensory system that seems to be fairly adept for a newborn infant is their auditory systems, or their ability to hear. And we're going to detail this more in a future class, but it is the one exception to these kind of poor sensory abilities that these children have at birth. So when we think about birth, when we think about this newborn, what we can probably recognize very easily here is that these kids really aren't born with much. They've got kind of this empty slate that's ready to pick up on all this information and be able to process things in the future, but they aren't really born with a lot of mechanisms at hand. In fact, what we usually see is that a lot of these children struggle with just kind of dealing with the daily stressors. And one of the common things that's actually done with these newborn infants is they're actually given tests to make sure that they can survive on their own outside of the womb. And one of the classic tests that's actually given is something that we call the APGAR test. And this is a test that's given to young infants. It's not a written test or anything like that. This is a very basic test that's given to young infants to see if, or I guess try to detect any problems that a newborn infant might encounter. So they test things like the heart, the respiratory system, muscular tone, things like body color, or even basic reflexes, in order to really look at whether or not a child is going to need kind of rudimentary help in order to be able to survive outside of the womb, or whether or not that child is kind of fine on its own. This kind of rudimentary check through the APGAR test or other tests that are done is done right at the moment of birth, so within a few minutes after birth, and then a few minutes later to make sure that the child is progressing in a normal stage and able to actually develop and kind of survive outside of the womb, as I mentioned, without any specific help. In addition, though, to testing kind of these basic stress reactions, what we also are looking for is the presence of very specific reflexes. We talked about some of these reflexes when we talked about early development, like the sucking reflex, or we called it the grasping reflex. And these particular reflexes, or actually we call it the plantar, I guess, but it's also called the grasping reflex. These particular reflexes should be present in young infants when they're born, and they might not be tested for all of these different reflexes immediately when the child is born, but within a very short period of time, most psychologists and most doctors will look for these reflexes to ensure the child's nervous system has grown successfully, because even though these children might not be able to voluntarily move their muscles the way they want to, there should be these kind of naturally inborn things that will help a child, at least theoretically, survive outside of the womb without having to process anything or make any decisions to react in specific ways. And what I'd like you to do, just so you can get a sense of some of these basic reflexes, is actually go online to this YouTube link that I created so you can see a video that looks at some of the very basic, very simple reflexes that young children are actually able to possess at birth, and which should be present if a child is going to be able to survive outside of the womb. Alright, hopefully you got a chance to check out that video. We're going to be taking a short break here now and moving on after this to that last section of abnormalities that can actually occur during the developmental process. So please, if you haven't, go see the video and then take a little bit of a break here so you can get yourself refreshed and ready to go for the last section of today's class. Welcome back. Now, as we were discussing in the first section, when we looked at kind of this idea of genetics and genomes, very often that genetic process unfolds perfectly, but in some rare cases, there are specific disorders that come out. We call these disorders congenital defects. And when we look at these congenital defects, approximately 1 in 20 individuals are born with something that makes them unique. And that uniqueness, that problem that can arise, can come from a number of different factors. One big chunk of those factors are called inherited defects or heritability disorders. The other set of defects, ones that we'll actually also discuss in this section, are ones that we attribute to the environment. Both of these processes, either inherited or environmental, can impact the child in a number of different ways. And what we're going to do in this section is kind of break down each of them. We're going to start off with inherited defects, and in particular move to a topic of chromosomal abnormalities in order to discuss exactly how some of these inherited defects can actually come about. When we're talking about chromosome-linked abnormalities, what we're actually looking at very often is problems that occur because of some mishap in the meiosis process that's actually occurring in the gametes that either the mother or the father is actually producing. And when this problem occurs with the meiosis, what happens is individuals are born with either too many or too few pieces of information of a chromosome that's critical. One of the more classic chromosome problems is one that we already foreshadowed earlier, that we call Down syndrome. And this particular disorder is highlighted by some very obvious physical indications. So we see the almond-shaped eyes, the protruding forehead, a number of other kind of classic things that your book talks about with the physical problems. But we also see a lot of mental and motor skill problems with these individuals. One of the classic terms that's often used for these individuals is mental retardation. That particular term, though might be controversial outside of a psychology class, is a very common used term that we use to explain kind of the slowing of the developmental processing, or the retarding of the mental process that these individuals actually encounter. This particular problem happens with about 1 in 700 births, and it's a problem that's related to a very specific chromosome. In particular, that 21st chromosome pair that we see has, for some odd reason, usually an extra copy of chromosome 21 within the mother. And when we look at this particular problem, what we often see is that this disorder of Down syndrome is very, very, very commonly linked to age. In fact, very few women who have a child between the age of 16 and 34 have a child with Down syndrome. But if a mother gets over the age of 34, or 35 and beyond age, or even younger than 16, we see a much higher prevalence of Down syndrome. And as mothers get older and older, into their 40s and 50s, when they bear children, their rates of having children with Down syndrome increases dramatically. And we believe that this is related and linked to kind of the slowing down or the decline of the crossover that we saw of the genetic material in this meiosis process. And we're not sure why this is the case or what's going on there, but we've definitely linked this kind of decline in the crossover to Down syndrome cases. Another very common chromosomal linked abnormality is something we call Klinefelter syndrome. And in Klinefelter syndrome, what we see is males with very underdeveloped testes and large breasts, and males that are very, very tall compared to other individuals. And this is a fairly rare chromosomal linked abnormality. About 1 in 100 births happen. But this is another one where we see something going on with this meiosis process. And for this case, what males have is an extra X chromosome, and that's obviously the 23rd chromosome pair that they have. So instead of being XY, they're XXY. So they have all the, I guess, biological makeup of a male, but they have an exceptional amount of kind of female characteristics to them. In fact, a large portion of their body and a large portion of them actually resembles a female more than a male. We have recently linked this particular syndrome to other mental and physical impairments as well, but there is a strong belief by a lot of psychologists that a lot of these impairments are actually due to more social problems. People are reacting to them differently because they have a different look to them, and really some of the challenges that they have with kind of dealing with these sex and identity issues and struggles that they very well could be encountering on a regular basis. Something that's very similar to Klinefelter syndrome is something we call Fragile X syndrome. And Fragile X syndrome actually occurs when both males and females have a very shortened, or I guess kind of fragile, as it's said, problem, or chromosomal structural problem with one of their X chromosomes on that 23rd chromosome pair. Now, for males, that Fragile X is extremely impactful because they only have one X chromosome, where with females, it's not quite as effective, not quite as pronounced. And in fact, males usually are identified with having Fragile X syndrome about 1 in 3,600 births, and females are only really identified as having Fragile X syndrome in 1 in 5,000 births. However, there's a belief that this kind of difference is based on the fact that it just doesn't seem to be as noticeable in females as it does in males. But when it happens, what we see in this case is individuals that suffer from things like short attention span, very, I guess, slow learning abilities. In fact, a lot of people with Fragile X syndrome have sometimes been misdiagnosed as having something that we call autism. We're going to be talking about autism a little bit later when we talk about attention span, but this particular disorder is something that's actually linked to chromosome problems, and it's very similar to Klinefelter syndrome. It's linked to that 23rd chromosome pair that individuals have. One final actually there's two more, sorry. One of the final chromosome abnormalities that's out there is something we call Turner syndrome. And this is when individuals only have one X chromosome. So for some reason, the male or the female, I guess, did not provide a second X chromosome. Now, there's a possibility that some offspring can only have one Y chromosome, but in this case, these individuals do not survive. However, with Turner syndrome, a large portion of these individuals do actually survive, even though they only have one X chromosome, or one chromosome. But when they are born, they have some very, very recognizable physical abnormalities. Very often they're very short. They have what's called a webbed neck, and you can actually see what a webbed neck looks like by looking at the girl in the right-hand corner of your slide, where their neck is kind of connected to, or their head, I guess, is connected to their shoulders. And usually these individuals have a couple cognitive things, but the only major other thing that we see with these individuals is that most of them are infertile because of their lacking that second chromosome. This is a very rare particular syndrome. In fact, only about 1 in 2,500 births actually involve children with Turner syndrome, but it is one that obviously has a big impact on these people's lives. A fourth chromosome abnormality linked to this 23rd chromosome is one that's a little bit unique because it's not one that's really a giant cause for concern. In fact, this is one that might be much, much more prevalent than we think, but it's not one that we usually identify, because most of the time what we see with this particular disorder is really kind of minimal changes to a couple characteristics in males. In fact, it makes males just more masculine, you would think. So they're much taller than most males. They have a tendency to be much more aggressive. And there's a slight impairment in their cognitive skills. However, these effects are very tough to identify, and unless researchers are really looking for something else, very often this particular XYY syndrome is actually missed. Right now, about 1 in 1,000 births are detected as having this chromosomal abnormality, but as I said, this might be something that's a little bit more common than we think. It's just not something that's identified nearly as much. Now, these chromosome abnormalities obviously are fairly often linked to that 23rd chromosome, but there are some other ones that are there. All of them involve either getting too much of a chromosome or too little of a specific chromosome. But the idea is that not only does this kind of impact kind of the genetic combination that people get, but it impacts a lot of the phenomes that a person's actually able to display because of the unique combination of chromosomes that a person gets. But these definitely aren't the only things that can impact individuals and can change the birth and their normal pattern of growth. One of the things that can also impact people's growth is specific genes that they're actually given from their parents. Some genetic abnormalities are due to what we call dominant genes or dominant alleles. Others are attributed to kind of recessive pairs or even codominant pairs. But when these individuals have these genetic abnormalities, what very often happens is that a child inherits kind of a disposition or a major physical impairment because of the specific genetic combination that they're receiving from their parents. And because we can actually detect a lot of these genetic abnormalities, unlike the ones we just talked about before, the chromosomal abnormalities, there's been a push within the community, in particular within the birthing community, to provide parents with something that we call genetic counseling. So if a parent knows that they have a specific allele that causes them to have a predisposition for specific things like depression or schizophrenia or any other major problem that they might encounter along the way, there are services that allow parents to kind of figure out exactly what their genetic combinations are and what the odds of producing offspring that could have these combinations in the future. In addition to that, this genetic counseling has also been kind of extended to kind of allowing parents to figure out whether or not they want to carry out a birth or whether or not they might have to do specific things if they do have a child. One classic example of the effectiveness of genetic counseling is through a disease or disorder that we call PKU. PKU is actually, it was actually in the past a disorder that was considered very life-threatening and very problematic. And when children were born with a specific genetic predisposition, they were born looking 100% healthy, but as they aged, very often their cognitive skills, their behavioral skills, and a lot of other things about them started to decline, and they weren't sure why this was the case. But we eventually detected this genetic marker, this genetic link, that kind of allowed researchers to predict when people were going to have this decline. And what we quickly realized after kind of detecting this was that one of the major problems was that these individuals couldn't process animal proteins, in particular any fats or any of the combinations of chemicals that are in most animal fats from meat and milks, and a couple of other grains that these people can't digest. And once we realized that and started kind of figuring out the triggers to the symptoms of PKU, we were actually able to kind of teach parents to have their children avoid these things. And dramatically, and this was very kind of intriguing and very, heartening to the researchers looking at this, once these parents stopped exposing their children to these things, these individuals actually developed normal lives. So that gradual decline that was linked to PKU never happened to the parents that didn't expose their children to these environmental factors. And that was a great example of an accomplishment that this genetic counseling has had. However, recent genetic counseling has looked more to different types of tests. So one of the tests that they call is amniocentesis, where a parent can actually have the genetic information of their child tested in order to determine a couple things, like whether or not they want to terminate that particular birth. And that idea of going in, testing your child, finding the genetic combination, and ending that child's life if the genetic combination isn't to your liking is something that's grown into a very controversial topic nowadays, because there's implications for whether or not people are just trying to have specific genetic combinations in their children instead of just avoiding genetic abnormalities. And another problem is that a lot of times these tests have the chance, not a great chance, but have the chance of actually kind of prematurely ending the life of these children. And because of that, there's a lot of heat in this particular field, but there's a lot of parents that also strongly believe that, they don't want a child to be born just to suffer for 2 or 3 years and die. And that's one of the major kind of counter complaints to this argument, and it's not something that I intend to solve in this class, or I intend you to solve in this class, but it is something that you should be aware of when we look at this topic of genetic abnormalities and how we can detect them and how we might be able to kind of counteract some of the problems that we've linked to these problems. In addition, though, to these biological things, so we've got chromosomal abnormalities and genetic abnormalities, another problem that very often comes up is that a lot of abnormalities at birth are due to environmental factors that children are exposed to inside the mother's womb. Now, I know a lot of you are thinking, well, the mother's womb doesn't change much, but in actuality, what happens to a child when they're connected to their mother, when they're tethered to them inside her womb, is that child is actually exposed to almost everything that the mother is actually exposed to. And some of those things in the environment can actually prove to be harmful to the child. And those things that are harmful to the child are labeled as something we call teratogens. And this is anything, any agent in the environment that can possibly have a negative impact on the child or even cause a birth defect. And when we look for teratogens in our environment, what we see is that there's a lot of things that, I guess, have to be taken into account when we're looking at these teratogens. One of the things that we have to take into account is how much of a teratogen somebody's actually exposed to. So if a child is exposed to only a small amount of one teratogen, it might have very harmful effects on that individual, where if they're only exposed to a small amount of another teratogen, you might not see any effect on the child whatsoever. And it's only when they're exposed to large amounts of that specific teratogen do we see any changes in the child's, I guess, birth outcome. In addition, though, to looking at these kind of exposure things, what we also see is that teratogens come in a huge variety of forms. Drugs are one of the more common ones that we see, but a number of other things that have started to pop up over the years have kind of moved away from drugs, like alcohol and drinking and other things, alcohol and caffeine and nicotine, and now have started to move to things that we never really considered as possible teratogens. So certain things that parents eat, in particular some fish and other objects, have now been considered teratogens, and there's different diseases that a mother can be exposed to, or even just different emotional states and encounters that a mother can have that actually can have an impact on the child. And what we're going to do is look at some of the more common teratogens that are out there. One of the more common teratogens that children encounter are ones that deal with drugs. Even though we've kind of moved away from them, they are definitely the more common ones that are out there. And the most common drug, the one that's gained a lot of interest and concern over the years, and I'm sure a lot of you have heard about this already before taking this class, is the teratogen of alcohol. And that's because alcohol can affect children in a number of different ways. It can impact limbs, how they develop. It can also impact facial abnormalities or even intelligence issues within children. And when we're looking for children who have been exposed to the teratogen of alcohol or experience something that we call fetal alcohol syndrome, there's a couple hallmarks that we're looking for. These individuals actually display facial, characteristics that are very similar to children that have Down syndrome. But there are some kind of small exceptions to this that really differentiate it from Down syndrome. One of the things that's kind of linked to fetal alcohol syndrome that's not linked to Down syndrome is a small head. Another thing that's kind of uncommon between them is kind of some of the folds within the nose, in particular the bridge of the nose, and the development of the jaw of these children. These abnormalities are things that are more physical and not something that's maybe a huge cause for concern for parents unless, looks are important. But another thing that's very, very problematic with this is that these individuals don't only experience specific facial feature problems, but very often there's a huge impact on their intelligence abilities as well. And this impact is predicated on how much alcohol a mother's actually exposed to. Some researchers believe that even a drop of alcohol can impact a child in a negative way, while others believe that it requires a decent amount of alcohol exposure to actually impact a child in any really severe way. As a psychologist and as somebody studying this, there's kind of a huge stress to say, there's no reason to ever be exposed to alcohol when you're bearing a child. But there is definitely a spectrum of effectiveness that we see with this particular disorder. Another one that's very similar in terms of kind of the cognitive impairments that alcohol have is the teratogen of nicotine that we've kind of come to recognize. And when we look at nicotine impacts, what we see very often parents who smoke produce children that have very low birth weights. And also, what we've started to recognize is that attention issues seem to be much more prevalent in children whose parents have been exposed to nicotine. And those cognitive impairments, sometimes also paired with some mild intelligence issues, have led a lot of researchers and parental supervisors to kind of avoid these major teratogens while the mother is caring for her child and while that child is in the mother's womb. Some other kind of random teratogen issues that we've recognized over the years deal with kind of things that are somewhat out of the control of the mother. One of the really interesting and very kind of scary teratogen issues deals with the mother's blood type. In particular, if a mother has a specific blood type that we call Rh negative, and the fetus has a blood type that we call Rh positive, this is problematic, not all the time, but in a number of instances. And that's because the mother's immune system actually will detect that child's blood as something that it needs to get rid of, as kind of something from the outside that needs to be removed from the mother's system. And that actually causes the mother's immune system to attack the fetus. And if the mother isn't treated, if her immune system isn't suppressed so she doesn't have her body attack the child's body, a number of really extremely negative factors can happen. Now, depending upon where this process occurs, we'll talk about this in a second, we can see that that mother's nervous system or that mother's immune system will actually attack the child in either the heart, brain, skin, or even a number of other categories that are out there. Another teratogen, one that's not very commonly linked, but something we've gained a lot of interest in over the years, is the maternal age of when the child is born. When a mother is very young, or when a mother is over the age of 35, not only do those chromosomal abnormalities occur that we talked about, but very often children are exposed to other problems as well. Low birth weight is a major problem especially with younger mothers, and mortality rates are very high with both young and more old mothers. And we see these things being related to either developmental processes, maybe the splitting of chromosomes in the meiosis stage, or just some of the exposures that these individuals actually encounter due to their age. When we're talking about teratogens, though, we've only covered a few of them. There's a number of them that are out there. And I'm sure you'll hear about them along the way. Things like being exposed to nitrogen or other problems that might be in the environment are very kind of common things that keep popping up time and time again. Instead of spending an entire semester, though, talking about these teratogens, what we do like to often reference is the fact that these exposure to teratogens have an impact on us in a number of ways, and it's usually dependent upon when that fetus is actually exposed to the teratogens. And that's because when a fetus is in the parent's womb, what's happening is that they're developing different regions of their body at different periods in time. And when that teratogen is, encountered, it changes which problems a child is actually having. So in the first couple months, say months 4 to 8, or actually the first couple weeks, weeks 4 to 8, these children are developing their hearts, in particular their heart system and all the valves that are important are really the main structural thing that's forming. And if a child encounters a teratogen, one especially that affects the heart, during that time, the child is susceptible to having heart problems. But if that child encounters that exact same teratogen later on, and it's specific to the heart, say at week 20, then that's not actually going to be something that's really problematic for the child. So when we look at teratogens, what we see is that they can have impact on a number of different things. Some of them carry over to a large system of different problems. Others are very specific to specific regions. And to know whether or not that teratogen is going to have an effect, we have to take into account how far along in the developmental process that fetus actually is. And we're going to talk more about this idea of these periods of growth later on, but I want you to kind of be familiar with this idea and this concept in order to understand the worry and the concern of teratogens when we talk about some of these genetic and other abnormalities that people experience at birth. That actually marks the end of today's class. Just to sum things up, we've covered a lot today, but what we were focusing on primarily was very, very early development. So we looked at the concept of heritability and genes, and then looked at development from a zygote to the moment of birth, and finally ended up talking about some of the problems, either environmental or genetic, that can sometimes occur during this early stage of development that can have long-lasting impacts on an individual, not only at the moment of birth, but throughout their life. If anything was confusing or anything was too much, please, by all means, stop, go back to the slide that you found challenging or confusing, and play it again. And if you have any other questions or confusions about it, still email me, let me know, and I'll be happy to try to clarify things for you. That's it for today, obviously. In the next class, we're going to be talking about development after birth. So we'll be looking at physical changes and other manifestations of that. And for that next class, your requirement, obviously, is to not only listen to the next slideshow presentation, but, as you're probably suspecting already, to read a chapter. And this time it's chapter 3. Please have that read by the time you listen to the presentation next week, or at least very, very soon after you listen to that presentation, and definitely before you submit that write-up. Get those write-ups in for this week on time. Please do not procrastinate. I look forward to hearing back from all of you about this particular presentation, and I wish you all the best in whatever you're doing this week, and hope to be speaking with you soon. Take care.